The topic for the day is decoding the mystery of embryo culture media. So if I have to design an embryo culture media, so what will be my thought process? So I'll look at how it functions and what it supports. So if I say that what it supports an embryo. So I have to look at the journey of embryo. Embryo is an unbound suspended organism, which and its pre-implantation development ranges from fertilization of oocyte to the hatched blastocyst stage during a course of around 5 to 7 days. Its nutrition depends on its own cytoplasmic stores of maternal origin in the beginning, then on the secretions of oviduct and later the uterus. So if you look at the figure below, it shows that the media should be able to support maturation, fertilization, cleavage, compaction, and ultimately cavitation, which will lead to implantation of the embryo. So if you look at what happens in the female genital tract, so that embryonic development, mainly it ranges from day 0 to day 3. It happens in the fallopian tubes, and then later the embryo moves on to the uterus on day 4 and day 5. So the composition of these secretions differs as the fertilized oocyte progresses from the fallopian tube and ultimately into the uterus. So when the scientists, they sat down and they thought, how do we design the media? They had to capture the small amounts of follicular tube, fallopian tube fluid as well as the uterine fluid, and then they had to decipher its secretions. We were discussing the ART journey. More than 8 million children have been born from IVF since the birth of Louis Brown in 1978. The great improvement in culture conditions, and especially the IVF culture media, is a very important part of this success. If you look at the history of embryo culture media, it all started with Ringer's media in 1882, which was the first tissue culture media and based on blood serum, and it was able to support a beating frog heart in vitro. But nowadays we've come to a time when the culture media constituents are up to 80, and it includes various growth factors, vitamins, nutrients, insulin growth factors, and so many other constituents. But the journey has been very complicated, and a lot of people have put a huge effort. I'll just touch upon the key developments. In 1956, Witten, who is known as the father of embryo culture media, he developed a media which could support mouse embryos up to blastocyst stage. Step 2, in 1971, he could culture human embryos up to blastocyst stage using Ham's F10 media with human or fetal calf serum. The first IVF baby was born in Earl's simple salt solution. And then in 1996, the landmark sequential media was developed by David Gardner and his colleagues. Now, there is a lot of controversy between sequential and single step. So we'll not touch upon those controversies, but we'll just touch everything briefly because time is also limited. Harper in 2012 said that despite so many developments happening and 150 years of research into this, but it still remains true that there is no culture media available that is truly optimized for human embryonic development. It is for the simple reason that it's not only the media which is important, it's about culture conditions, it's about the embryologists, it's about so many other things, and we really can't simulate the natural conditions absolutely 100%. So when you look at the embryo culture system, the primary functions are it should be able to support the viability of gametes, sperm functions, oocyte maturation, oocyte competence. The embryonic stress should be minimal, so that there should be very strict and tight control over temperature, pH, CO2. Embryonic development should be normal, that is fertilization, genomic activation, compaction, cavitation, all the steps should be optimally supported. Only then good pregnancy rate. So the embryo culture system, it has got a few components. First is culture media, which we are going to discuss, as well as consumables, culture conditions, quality control, culture which vary from lab to lab and conditions to conditions. And ultimately, it's the embryologist who is taking care of all these constituents and trying to use them optimally to give excellent pregnancy rates. This is from David Gardner's book, in which he has just outlined so many variables which are responsible for the ultimate outcome. It depends on patient factors, lab factors, number of embryologists, how well they are trained, number of incubators, air quality, oil overlay. I mean, there is no end to the variables. So that is the reason why we have not been able to obtain optimal results in all the labs or in most of the labs. So now coming to the topic for the day, decoding the culture media. If you look at the culture media, the constituents are the basic backbone is the same as any other media. It has got salts, sodium, potassium chloride, calcium chloride, energy substrates, which we'll touch in detail, protein. There's a complex mixture of amino acids with glutamine in the form of dipeptide. Phenol red as a pH indicator, which is again optional and not all medias have it. Antibiotics are part of all the medias, and generally you will find, if you look at the constituents, it's penicillin, streptomycin, and gentamicin. These are the most commonly used antibiotics. Energy substrates, mainly three: pyruvate, lactate, and glucose. Pre-compaction embryos, they always prefer pyruvate. They prefer pyruvate, followed by shift to glucose post-compaction and blastocyst formation. Most culture media provide pyruvate at the same concentration as in fallopian tubes, that is between 0.2 to 0.5 millimoles per liter. Amino acids and EDTA, they are also a source of energy, and with the additional function that they limit the utilization of glucose during pre-compaction stage. So the glucose has to be limited. Pyruvate is promoted as a pre-compaction energy substrate. Whereas if you look at the post-compaction embryos, they prefer glucose as an energy source, the physiological level of 3 millimoles per liter in blastocyst stage medias. Amino acids, they are, like I discussed in the previous slide, they are a source of energy, and they act as osmolytes, buffers of intracellular pH, antioxidant chelators, signaling. The inclusion of amino acids in the culture media, it enhances the embryonic development up to the blastocyst stage. And if we kind of see which amino acids are optimally used in which stage of development, if you look at the cleavage stage, non-essential amino acids form the bulk of amino acids. They act as strong intracellular buffers of pH due to their zwitteronic nature and chelate toxins. Compaction amino acids, both non-essential as well as essential amino acids are present in these medias. Non-essential amino acids and glutamine, it increases the blastocyst formation and hatching, whereas essential amino acids stimulate cleavage rates and increase development of the ICM, or the inner cell mass. It was seen that the addition of glutamine to a simple culture media significantly increased development of human blastocysts in culture and increased the pregnancy rates. So that means glutamine is a very important component, but then it comes with its own drawbacks. L-glutamine, an amino acid with a very vital role in embryonic development, it breaks down spontaneously with ammonium as a byproduct. And the impaction of ammonium in culture media is a major concern. Its effect is magnified in 20% oxygen compared to 5% oxygen. It alters embryonic differentiation and significantly reduces implantation and fetal development rates. So easy remedy was found, the introduction of glutamine in stable dipeptide form. It doesn't break down at 37 degrees, and modern day all the medias have glutamine in its stable dipeptide form in place of L-glutamine, which was used initially. Another group of components is chelators, such as EDTA. It chelates potentially toxic metals. It may be involved with glucose metabolism. As I told you in the previous slides, it limits the utilization of glucose, and it stimulates development prior to compaction. The presence of EDTA for blastocyst development compromises the subsequent development of the embryo. So generally, the post-compaction media, as they do not have EDTA, it increased the incidence of monozygotic twins derived from blastocyst culture in the presence of EDTA, which exhibit loose inner cell mass, and this is another drawback of using EDTA. Nowadays, a lot of medias are coming. Marketing people will come and they'll tell you, say, it has got insulin, it has got growth factors, it has got hyaluronan. But then you have to understand whether it is actually making a difference, or there is good quality evidence which favors the utilization of these substrates in the media. Like hyaluronan, it has got equivocal benefits for embryo transfer media. There are limited clinical benefits, and the benefit was mostly seen in females who are more than 35 years of age, who had very, very poor quality blastocysts available for transfer, and in whom there was evidence of previous implantation failures. But again, we also tried using the hyaluronate medias, and it actually doesn't make a big difference. Insulin, it improves embryo development and ongoing pregnancy rates, but further studies are needed. Vitamins B, C, antioxidants, all the media are containing antioxidants, such as lipoic acid, choline, ascorbate. But again, you have to understand that these antioxidants are beneficial only in certain conditions. They're beneficial in all the conditions, but then they are significantly beneficial in some conditions. Like this study by David Gardner, he showed that antioxidants are critical in single cultures at 20% atmospheric oxygen, where they're not very significant in 5% oxygen, and especially with the single culture. The group cultures, 5% they're not significant, whereas single cultures at 20% atmospheric oxygen, they make a significant difference. So when you're designing your culture system, you look at the incubators, what atmospheric oxygen it is offering, and accordingly you decide which media you have to choose. Nowadays, there are a lot of media available which contain GM-CSF, which is a multifunctional cytokine present in the oviduct and uterine epithelium. A large RCT showed a benefit of GM-CSF in very selected group of population, but only when the HSA was reduced. So there is insufficient evidence to support the addition of bioactive substances such as growth factors and complex proteins. So you have to, and these media actually are very expensive. So you have to weigh your risk and benefits and then choose your media. If you look at the buffer systems should be capable of maintaining a physiological pH between 7.2 to 7.4 in the media. They are selected based on their pK values, and the simultaneous use of multiple buffers allows for adjustment of pK values to the desired range, reduction in individual buffer concentrations, and potentially elevates concerns for toxicity. Generally, it is a combination of bicarbonate, HEPs, and MOPs. Primarily, if you are using it inside the incubator, is bicarbonate, but some amount of HEPs or MOPs is definitely present in all the media. And if you're using it primarily outside the incubator, then HEPs and MOPs are the primary constituents for buffer. Advantage of bicarbonate or CO2 buffer system is that it is the physiological buffer system present in the mammalian cells. A major drawback is the rapid pH change which occurs when medium is exposed to air, resulting in impaired development or even cellular necrosis if prolonged. A very simple solution is an oil overlay, which reduces gas exchange. Yesterday there was a talk by Dr. Joe. He said that if you have a reasonably good oil overlay, then the pH is maintained for up to 10 minutes. So that was a new fact for me, because I assumed that I have to work very quickly and I have to work within 2 minutes only. But he said that pH doesn't change with an oil overlay, it stays the same for up to 10 minutes if you're maintaining the right temperature. So you can also replace 20 nanomoles bicarbonate with HEPs, like I told you in the previous slide, leaving 5 millimole of bicarbonate. This also helps in maintaining the pH for a longer time. Such buffering system does not require a CO2 environment and can be used for short-term incubation of 10 minutes, less than 10 minutes. Phenol red, when we started our embryology practice, a lot of medias were available, still they are available, which offer phenol red, and we used to look at the color of phenol red for looking at the pH equilibration in the morning. It generally turned from purple to light pink, but nowadays in the era of microdroplet culture, I don't think we can really appreciate color between light pink and a transparent media. Plus, if phenol red does have some hypothetical risk based on its estrogenic activity, source of ROS. It's a source of ROS, and it has got potentially no role in embryonic development. So phenol red, if it is not present, maybe I think it is not making a significant difference in terms of quality control nowadays. The discussion cannot end without a discussion of the oil. An oil overlay is essential, because then you can culture in reduced volumes. You can do a single culture or a group culture in a volume of around 20 to 50 microliters. It prevents the evaporation of media, reduces harmful effects such as increase in osmolality, reduction in pH, or changes in pH. It prevents the risk of temperature fluctuation, and it has also an advantage in trapping a lot of VOCs. So generally we recommend that maximum amount of oil should be used in a culture dish. Generally, two types of oil are available: paraffin oil and silicone oil. Paraffin oil is same as the mineral oil, and they have got their own set of advantages and disadvantages. But certain things are common to both kinds of oil. They should be stored in dark and in glass, preferably. It should not be stored for extended periods in the incubator. It should never be coated into tissue culture flask, as they are styrene based, and oil is able to leach styrene from such containers at very high rates over time. Oil is susceptible to photo-oxidation or peroxidation with accumulation of peroxides that are damaging to embryo culture. In the presence of elevated peroxide levels, albumin in tissue culture media can facilitate the transfer of free radicals across zona pellucida, leading to, possibly due to the capture of free radicals by free sulfidyl groups in the albumin molecule. Now, coming to the storage of media, or generally media handling, ensure that the cold chain is not broken during transfer of culture media between manufacturer, supplier, and the clinic. You should always keep the media in dark, avoid light, as two of the most labile components are amino acids and vitamins. Glutamine is the most labile amino acid, and it produces highest levels of ammonium of any amino acid. Store the media continuously at the appropriate temperature. Refrigerator designed for medical storage with transparent sliding doors should be preferred over normal kitchen refrigerators. Media is sometimes allocated into very small volumes after delivery. This should be avoided as it releases VOCs. Media should be incubated for temperature and pH 4 to 6 hours before use, or by overnight incubation preferably. It should be placed in the incubator for the minimum time required for incubation, and certainly they should never be stored in the incubator. A brief discussion about the culture protocols. We'll just touch upon as sequential versus single, open versus droplet, and humidified culture. Now, there are two approaches. I'm not going to go into the details. One is the back to nature approach, and the other is the let the embryo choose approach. Back to nature is the sequential, which I discussed in detail at the start of the discussion. And let the embryo choose is a single step, which is nowadays very common, especially with people who are using time lapse. So if you look at the single versus sequential versus single debate, in single media, if you're not renewing the media, the embryos are left undisturbed. So a lot of toxins can accumulate, whereas the positive side is that endogenous growth factors and autocrine and paracrine factors are also left undisturbed. If you renew the media, single-step media, then I don't think there is much difference between using a single-step media with renewal compared to sequential media, because in both the endogenous growth factors are lost. The toxins are removed. That's a positive thing, and obviously the single-step media, it requires less degree of quality control as you're handling just one media, so the chances of error are reduced. But that is not something which is very important because that can be taken care of if you work diligently. If you look at the evidence, a recent Cochrane review concludes that there are no differences in outcomes between two culture systems, and the conclusion is supported by many systemic reviews. Although using a single-step media for extended culture has some practical advantages and blastocyst formation repair rates appear to be higher, there is insufficient evidence to recommend either sequential or single-step media as being superior for the culture of embryos. So the gist is that there is insufficient evidence. You might be able to obtain higher blastocyst and higher number of cleavage stage embryos, but ultimately the live birth rates are not changing much. If you look at the humidified versus dry culture, some of us have incubators which are dry. Some incubators do, the gas is the gas comes with water vapor. It's a humidified incubator. So recent studies have shown that the oil overlay is definitely insufficient to protect against osmolality changes during dry incubation, especially if the incubation is prolonged. And the effect of changes in osmolality in non-humidified culture may be less deleterious in culture with regular medium renewal. So either you culture in humidified incubators, or if you're culturing in dry incubators, you have to renew the media. So if you're renewing the media, so the thought is that why not use a sequential media? So specific culture conditions such as oil volume depth, medium surface area to oil interface, ratio of oil to medium, room humidity, pre-humidification of oil, medium droplet size, as well as medium changes can affect individual results. Now you can either culture in an open dish, like there's a single well dish, you put in media, put in oil, that's your choice. Oil is again optional. It was the dish in which Edwards was culturing till almost 1981. Evaporation is obviously greater in open cultures, causing a lot of osmotic changes in the media. The chances for contamination by microorganisms is high in such dishes. The large volume of medium used may dilute metabolites in paracrine and autocrine factors. Obviously, this is like fish swimming in an ocean, and so obviously even if they are releasing some factors, they can't reach the other embryo.
Sarabpreet, we are running late. Please.
Just last slide. pH will is going to change due to loss of CO2. If you look at embryo culture under oil, in this it eliminates loss of embryos during handling, minimizes the chances of microbial contamination, it absorbs the VOCs, and maintains the metabolites and paracrine and autocrine factors. I'm not going to the details, and again, the last topic I'm going to touch is single versus a group culture. In group culture, the benefit is in autocrine and paracrine factors, and the optimal density is definitely not known till now. Some people recommend 5 embryos in 50 microliters. Some recommend 4 embryos in 20 microliters. So generally the consensus is that you can culture up to 4 human embryos in 50 microliter drops, and this maintains all the benefits of droplet culture as well as a group culture. Thank you.